Yeah, welcome everyone. This is the fourth webinar of PDB's API webinar series. And I am Srinath Nair from the development team at PDB. So here I'll be discussing about the PDB Graph API, which is also referred to as PDB Aggregated API. As I mentioned before, this is part of our API webinar series. We have 2 more webinars and in this series to come in next few weeks. Just giving a brief idea on what are the things that I'll be discussing today. First, I'll start with the types of data that we have in PDBe. Then I will discuss a bit about PDB graph database, how to download it. Then I will take you to the PDB graph API and where are we using it within PDBe. And at last, we'll go through a few use cases where it could help you in getting the data. PDB data is managed by many sites, which comes under Worldwide Protein Data Bank. And ourselves, PDBe is a founding member of Worldwide Protein Data Bank and archives experimentally determined tree structures of biological macromolecules. In addition to protein structures, we have nucleic acid structures and small molecules as well. We are based in UK along with the EMDB. We also have RCSP PDB and BMRB, which are based in US. And in Japan, we have PDBJ, which is another site who manages PDB data. And all the data in Protein Data Bank is deposited using WWPDB's deposition system named OneDep. The coordinate data as well as some additional data are received from the depositors. These data are enriched as part of the curation process. This includes ligand processing, sequence processing, where PDB sequence are mapped to the UniProt sequence, and also other value-added annotations. Then each structure goes through a set of standard validation pipelines. These are all available in the archive, where you can see MMC files which can be used to get the coordinate data as well as other information. There are also validation reports which are available as validation XMLs. We are at the heart of European Bioinformatics Institute, surrounded by a host of useful resources. We use these resources to link structural data that we have with other functional annotations like sequence annotations, variation data, chemistry data, etc. This made us initiate many projects which integrates various of these resources to come up with many useful annotations. Pdbkb is a community-driven integrated resource with more than 20 collaborating partners. The motivation behind Pdbkb is to create an infrastructure for structural and functional annotations of PDB entries based on FAIR principles. Pdbkb integrates structural and functional annotations of PDB entries on residue level from Pdbkb partners. Some of these annotations include predicted and curated ligand binding sites, catalytic sites, PTM sites, and more. So we have the core PDB data here. Then we get annotations for PDB entries from specialist databases, which gets into an integrated graph database. And then we use this data to be exposed on the aggregated views. I will talk I will talk about it in the later slides. Thus, by integrating Pdbkb data with the core Pdb data helps in answering some specific scientific questions. SIFT is one of the collaborative project with Uniprot. It stands for Structure Integration with Function Taxonomy and Sequence. This project aims in linking the structural data in PDB to the sequence data in Uniprot. This helps us in integrating more annotations. For example, gene ontology, sequence families from Interpro and PFAM, structural families from SCOPE and CAT. So what is a graph database? A graph database is a database which is designed to store and retrieve data in the form of a graph. So it treats the relationships as equally important to the data itself. The graph database uses nodes to represent an entity and edges to create connection between them. PDB data itself is inherently a tree-like structure. So you can assume each PDB entry as a node, which are connected to the PDB chains and entities, which are later connected to the PDB residue nodes. We have diverse types of residue level annotations. So we have connections. So we have made connections from the PDB residue nodes to many other annotations like this. To solve some of the scientific questions, it would be essential to retrieve different types of these annotations, which could easily be done using a graph database. It also helps us in finding some interesting patterns, which could help us in getting some valuable information. The residue level mapping with the Uniprot helps us in the straightforward transfer of these annotations between the PDB entries and the mapped Uniprot accession. If you want to download the PDB graph database, you can use this URL below. So how do we make use of this data? Programmatic access is one way of using PDB data to gather this rich amount of information, and it can be used to incorporate them in your pipelines. We basically provide three offerings in terms of programmatic access to PDB data. You can use this link to access this homepage of our API. So the first kind of API that we have is based on Apache Solr. It's a query system which provides an advanced search capabilities across 200 fields. This API is used in our advanced search components, like in the entry pages and the aggregated views. So the second one is an entry-based API, which relies on our relational database to fetch details for a PDB entry or some ligand IDs. The third one is aggregated API, which is also referred to as Graph API. It is a newer the API that we have in PDBe, which is powered by the PDB Graph database. We'll be talking about more in about aggregated API today. So the PDB Aggregated API or Graph API is a RESTful API powered by PDB Graph database. This provides the API calls for aggregated data from across relevant PDB entries. For example, based on entities, Uniprot IDs, residues, and more. It currently serves close to about 80 endpoints, and it supports and only GET request currently. So let's talk about the documentation page of API. This is a web-based documentation where you can get details of all endpoints exposed by Aggregated API. There are two main components in the documentation. So one is the sidebar on the left side. Here the API endpoints are listed and put into different categories for easy navigation. It is also equipped with a filter text box on the top to find an endpoint of interest. For example, if you're looking for interface residues for a protein, you can search for interface in the text box, which will give you the particular result that are based on the keyword interface. Then once you find the API endpoint which you are looking for, you can click on it and it will take you to the actual documentation of the endpoint. So this is the documentation page for the interface residues API call for a unit accession. And this particular section actually gives you some description on the type of endpoint that we have, and also provides you with a URL which can be used to access this endpoint. It also provides you the information of the parameters to be used and the description of the fields, like in the result dataset in case of a successful response. There is also provision to try out the API from the documentation itself. For that, you can provide the word or the keyword which you would be looking for. So in this case, it's a uniport accession. And once you provide that in the text field, you can click on the send button, which gives you the response in the response, the text area here down. As I mentioned before, the endpoints are categorized for easy navigation. Let's go through these categories and see what data they can offer. So first, the compounds category. It provides data related to a small molecule. These endpoints can be used to get the summary information for ligand, atoms and bonds information, the PDB entries which bound to a particular ligand, etc. One useful endpoint in the compounds category is a similar ligands call. This API can be used to retrieve the stereoisomers, ligands with the same scaffold, and ligands with a similarity over 60% for a particular ligand of interest. This particular example is about the two similar antibiotics, the erythromycin and the clarithromycin, which shares the same scaffold. So once you provide the head code of the ligand, the response will be rendered, and there is a same scaffold field which can be used to get the ligands which have the same scaffold as that of the ligand in question. So next is the PDB category, which has endpoints that provide information for a PDB entry or an entity. These endpoints provide provide a variety of information like functional annotations from the Pdbkb partners, sequence and structural domains, bound ligand interactions from the RPGO, macromolecular interactions from PISA. There are also information of secondary structures, sequence conservation, residue modifications and mutations. And next is the residue section, where all endpoints that are related to a PDB residue or a range of residues are available. One useful API call here is the annotations endpoint for a PDB residue range. This API call can be used to get all the annotations for a range of PDB residues. These annotations include sequence and structural annotations, as well as functional annotations from Pdbkb partners. So to use this, use the parameters section to get the API result of the documentation. So once you fill in all the properties, like all the parameters, like which PDB entry that you're looking for and which entity ID, and the residues range from start and end, the result will be rendered below, or you can use the full URL like in a browser to get the result. The response for this API call is provided in a hierarchical manner, where you can find various information for a residue. So it starts with the PDB entry, and then it goes down to the entity and to the chains and to each residues. And these annotations for a residue are provided in the features field. So now inside the features field, each type of annotation have its own space. For example, uniprot, and each uniprot accession here, here, and here, and its corresponding annotations. Its corresponding residue mapping can be seen for them. So similarly, we have other mappings like pfarm, interpro, cat, scope, etc. And also the annotations from the Pdbkb partners can be retrieved from the fun PDB field. So for each of the partner, you can actually see the origin, the URL to the particular resource, the different scores like the row score, the confidence score, etc. Moving on to the next category, SIFs, where different endpoints provide data related to mappings as defined by the SIFs process. So you can get mappings to the uniprot, interpro, pfarm, sequence and structural domains, as well as uniref 90 cluster mappings. One useful endpoint here is to get the best structures for a uniprot accession. This API call lists down the PDB structures based on the coverage and resolution for a uniprot accession. So this particular example is for the replicase polyprotein found in the coronavirus. The results is a list of PDB, is a list of best PDB structures which are sorted by coverage and resolution. So this is a full URL which can be used to access the API call, and if you want to try out from the API documentation, you can provide the accession here, and the response will be rendered here. And all these PDB structures are sorted so that whatever PDB structure that you get that you can find on the top is the best. Now in this list you can find the details of like each entry like the experimental method, what is the resolution, what is the coverage for that particular segment, etc. So there's also another endpoint which gives the same information, but for a uniprot residue range. So you can use this URL and you can provide the accession and the uniprot start and end. And next is the uniprot category, which provides information for a uniprot accession. Most of these endpoints provides aggregated data across the relevant PDB entries, which are mapped to the uniprot accession in the query. We'll see examples of some of these endpoints in the later slide. And at last, we have the validation category. This has a list of endpoints which provides information that is related to the experiment and validation for a PDB structure. This includes calls like the Ramachandran stats for a PDB entry, or list of outlier types for a PDB residue, etc. So how do we make use of the aggregated API within PDBe? So with the power of highly interconnected data that we have in the PDB graph database, we could look at this data from different perspectives. This made us to come up with aggregated views, and we started making the aggregated views of proteins. So this is the home page of the aggregated views of proteins. You can use this link to access this home page. So aggregated views of proteins gives an overview of all the structural data that is available for a protein of interest. You can search for a PDB entry ID or a uniprot accession. So if you're interested in a particular PDB ID or uniprot, you can search for it from this text field. Next, I'll take you through some of these components and the corresponding API calls which are used to render this information. So this is how the aggregated views of proteins look like. So this page is for the Krebs binding protein found in human. There are many components used in this page, which is powered by the aggregated API, which aggregates data across the PDB entries for this uniprot accession, and provides the data in a very meaningful way. So there are few components here like this, which gives you the representative structures, and this is a summary component. So these components gives you a higher level of information as in here, and there are also components which are below, which gives you a more detailed information. Next, we will see some of these components and respective API like endpoints which are used. First, we will see the representative structures for a uniprot accession. So this component on the right side. This gives you the information on the non-overlapping PDB chains with the highest number of observed residues in the protein. The API call used for getting this information is the one here, which you can find from the uniprot category. So in the uniprot category, in the API documentation, you can find this API endpoint, get non-overlapping base structures for uniprot accession. This is a full URL which can be used to access this API endpoint, and you can input and you can give the accession, the uniprot accession, which will give you the API result. Moving on to another component which provides information on interface residues in a protein. It can be accessed from the aggregated view by clicking any of these. So this is an example of thrombin. So this says there are 48 interactions, and there is also a tab here on top, which also gives you the same information. And once you click on any of these links, it will take you to the molecular interaction section. And the molecular interaction section contains another component which gives you information on the interface residues. This displays each interacting partner in a row and highlights the interacting residues. So in this component, you can see there are interacting partners in a row. So each row is for each interacting partner, and there are some residues. So there are some residues highlighted in the position. So this is 1 to 622. So it means it's a full sequence for that particular uniprot. And we can see some of the residues at this location is actually interacting, and whatever we are highlighting here are the residues which are part of the protein-protein interaction. This data is provided by the API get interface residues for a uniprot accession, which can be found in the uniprot category.
So more details on this API call. You can use this following URL to access the endpoint and provide the uniprot accession. This will give you the result as seen below, and it also provides a basic details of the uniprot accession that you have given, like the sequence and the length. And the interacting partner details can be found in the data field, where you can see each interacting partner and its corresponding residues.
And another useful section on aggregated view is the ligand section, which can be accessed by clicking any of these. So in the summary section, it says there are 303 ligands, and there's a tab also on top which also says the same. The ligand section can be accessed by clicking any of these, which takes you to the ligand binding residues component. So this is a component which gives you the information on the ligand binding residues, which is similar to the interface residue section. We saw that in the previous slide. This component shows the ligand binding residues for a uniprot accession. And here you can see each ligand in a row, and the highlighted are the residues. So there are each ligand in each row, and there are residues that are so there are highlighted residues which takes part in the particular interaction. The API call which provides this data is the one here in the uniprot category, get ligand binding residues for uniprot accession. This API call also takes a uniprot accession and provides the data. So this is a full URL which can be used to access this endpoint, and it accepts a uniprot accession. As you have seen before, also it provides a basic information about the protein with the sequence and the length, and also in the data field data array it has a list of each bound ligand with its title record and the name, and also the residue. So the structure of it is very similar to the one that we have seen for the interface residues. So looking at both the ligand binding sites and interface residues data together shows some residues involved in both ligand binding and the macromolecular interactions, which are non-inhibitors. So this example is for the same example of the thrombin, what we have seen before, which functions in blood homeostasis, like inflammation and the wound healing. Another place where we use aggregated API is in the ligand environment, which can be accessed by clicking on the bound ligand in an entry page. So for each entry, for each PDB entry, there's an entry page, and you can find the ligands and environment section for each entry, and which gives you information on the ligands that are bound. So the ligand environment component can be accessed by clicking on this ligand, like on this bound ligand, that image that we have. So this is the ligand environment component, which is an interactive display of the bound molecules and their environment. The given API, which is here, get bound ligand interactions, which can be found in the Pdb category, is used to fetch the atom level interactions of the ligands in a bound molecule. So as you can see here, this is the full URL, and it accepts a PDB ID, a chain ID, and other sequence number, and it gives you the respective data for the query that is passed. So now let's see a use case. If you have a structure and want to know the residues which are accessible, you can make use of the annotations API call for a PDB entry for a particular resource. And this data is provided by the Pdbkb partner 3D Complex. So you can use the origin. So this is the full URL for it, and in the origin you can give the option called 3D Complex and the PDB structure that you're looking for. And this gives you the result. And in the result data set, you will be able to see the label and the row score. So you can filter the result on the label as ASA alone and the row scores which are greater than 0, which gives you the relevant residues which are accessible as defined by the 3D complex. Let's see another use case. And imatinib is a tyrosine kinase inhibitor for cancer. To find the ligands which are similar to STI, which can be easily found by using the similar ligands API code that is available in the compounds category. I think we have seen this before for the example of the erythromycin and clarithromycin. So this is the URL which full URL which can be used to access the endpoint, and you can pass a head code of your ligand, which gives you the result. And in the result, there is a similar ligands field, which gives a list of the ligands, including its ChemComp ID and the full name, and also the similarity score as defined by the parity method. We have established a Git doc to make available the materials like this tutorial slides and videos related to this API webinar series. To access that, you can make use of this short URL. Also, we will be adding a use cases section in the API webinar documentation, where you could find the worked out examples of the use cases that we discussed today, like as well as some others. So coming to the end of this webinar, in the next webinar happening next week, my colleague Lukash will be talking about the Pdb tools in GitHub. These are some Python software packages that are available, which can be accessed publicly and reused for your purposes. And use the links provided to get more information on the coming webinars. Please note that separate sign up is required for these webinars. So please make sure you register for them if you're interested. So thanks for listening to this webinar. I would like to stop here and open the session for questions if you have any.